The views expressed by me and you are our own. They don't necessarily reflect the views of CAUK or of current clinical advice. And that's applicable in this talk. And if you want to take advice, you must speak to your own clinician. So this is the fourth in a series of webinars on the science of Cavernoma, meant to be for the non-scientist. This one has changed from the one that I anticipated doing because a really important publication came out about a couple of weeks ago. Well, it announced that it was coming out and the preprint was available. So this webinar discusses a major new piece of research into the trigger for the rapid growth of Cavernoma that can result in hemorrhagic stroke and seizures. And this has been elusive because I've given talks where we've talked about the bleeding in Cavernoma, and people say, well, is that a slow bleed or a fast bleed? And I haven't known the answer. But this is talking about the bleeds that we talk about that are such a problem for Cavernoma. And as an acknowledgement, I've drawn on materials from the paper, from some of my own webinars, which those of you who've been before will recognize, but also on a very recent article on the Angioma Alliance website that was written, I think, by Amy Akers and Connie Lee. And that was published on the Angioma website, I think late last night or maybe this morning. And some of what I'm going to talk about is directly from there. This is the article that has been of such interest. This is, it says, something or other article preview. It's not all that often that Nature does this. This is a publication in a journal called, an article in a journal called Nature. And I've put it up here really to show that there are an awful lot of people who've contributed to it from 3 different labs in the States. But interestingly, the senior one is this person, Mark Kahn. Mark Kahn, those of you who came to the talk I did last month, excuse me, which was on the microbiome. He was the person who did that work, I mean, not by himself, with a lot of the people here as well. And he's been responsible for this major piece of research, which is going to be the start of something I think done quite a lot in Cavernoma in the future. Just one slide of sort of the basics. Cavernomas sit on the blood capillaries in the brain or spinal cord. Blood comes from the heart, round through the blood capillaries, out through the veins, back to the heart, and passes through the Cavernoma when the Cavernoma is there. And the blood capillaries are thin tubes. They have basically a single layer of cells lining them. These cells are what are called endothelial cells, part of the blood capillary. It's a fairly weak structure. It's small. It's not that much bigger than the diameter of a red blood corpuscle, and there's a sheath around it to give it some rigidity. And there's quite a lot of other cells around it which aren't relevant to this talk, and I haven't put in here. The diagrams I show are highly schematic. They don't, well, this represents the blood capillary moderately well, but some of the diagrams I have are just simply diagrammatic. Now, if you're one of the 99.9% of the population, this is a recap as well, then your blood capillary cells here are linked by tight junctions to the adjacent cells. And those tight junctions are a protein link that links from one cell to the other, and it prevents the passage of red blood cells, certainly, but also many other things from the inside of the blood capillary into the brain. And tight junctions are what form the blood-brain barrier, which I'm sure you will have heard about. But if you're one of the 0.1% or so of people who develop Cavernoma, then where you have the Cavernoma, you've lost those tight junctions. So that's represented here by no little black mark between the two cells. And at that point, the blood capillary becomes leaky. Blood corpuscles can often leak out, and certainly other things as well. And so in this region, you've lost the protection of the blood-brain barrier. This is only in the place where you have the Cavernoma. If you have a Cavernoma, in the rest of your blood capillaries, you still have the tight junctions. So it's only where the Cavernoma has formed that those are lost. Now, a lot of this talk depends upon this slide here, so I shall go through this. in a little bit of detail. If you're one of the 99.9%, these sort of grey things are diagrammatic chromosomes, and the white bars are genes. And you're familiar with the fact that we have 3 so-called Cavernoma genes, and those genes can exist in many forms. In the form that you have, if you're one of the 99.9%, they're in what I call a tight junction form. And the tight junction form means that you develop tight junctions and you have a normal blood capillary. About 0.1% of the population, not getting too particular about the numbers, inherit a CCM gene, a mutant tight junction gene, from one of their parents, and a tight junction gene from the other. People who, if ever there were any who had both parents with the Cavernoma gene, the fetuses would die very early on. So people who are going to develop familial Cavernoma have one mutant CCM gene and one tight junction gene. But that isn't sufficient to enable the blood capillary to form a normal capillary. Just having one of these tight junction genes is enough to develop the tight junctions. In order to be able to develop a Cavernoma, both the genes have to be the CCM mutant form. And so we have a problem, ostensibly, because we are born with either 2 tight junction genes or 1, but that isn't sufficient to cause a Cavernoma. Well, you might say, thank heavens. In order to get a Cavernoma, you have to have both of those. So how do you go from having a tight junction gene or 2 tight junction genes to the Cavernoma capable form of the cell? And the answer is what is called a somatic mutation. I shall talk a little bit more about somatic changes in a moment, but a somatic mutation is something that happens during your lifetime, during your childhood or adult lifetime, that enables the tight junction gene to change and mutate to the CCM gene. And somatic mutations are caused by radiation, by chemicals, by many, many things. If you have just one tight junction gene, you only have to have one somatic mutation. But if you have this normal situation that you have up there, excuse me, I've lost the pointer, that you have up here, then you have to have 2 hits, 2 somatic mutations, because both of these tight junction genes have to become mutated to the CCM form, enabling a Cavernoma. A Cavernoma doesn't necessarily happen, we'll come to that, but you can't get a Cavernoma without that happening. So if you're one of the 99.9% people who are born with 2 tight junction genes, you have to have 2 hits, and then you have, if you develop a Cavernoma, what's called a sporadic Cavernoma. But if you're born with the familial form where you have one CCM mutant from one of your parents, then, and you develop a Cavernoma, you develop what's known as a familial Cavernoma. And one final bit of terminology, and that is that this is known as a loss of function process, because the normal form allows or enables, helps develop tight junctions, and when you lose that function, you get the CCM mutants and you get the ability to form a Cavernoma. I said I'd talk just a little bit about somatic mutations, and here they are. So mutations, somatic mutations happen in cells of the body other than the eggs or the sperm. They're not inherited and they can't be passed on to your children. So if you develop a Cavernoma from this 99.9%, it's yours alone, you don't pass it on. And somatic mutations happen throughout your life. Taking all the cells of your body together, they're really pretty common. But then you have 30 trillion cells in your body. You have 4 billion blood capillary cells. The total length of blood capillaries in your brain is in the region of 50,000 miles, or it's 80,000 kilometers for those who don't recognize miles. That's an incredible length. I like numbers, that's my problem. Now, these 2 hits that we talk about have to happen in a single cell, and they have to happen on the adjacent genes that are the so-called Cavernoma genes. And so you can get a somatic mutation in one blood capillary cell, and then another one in somewhere else, and another one in somewhere else. And that doesn't help or hinder, if you like, the ability to get a cavernoma. It has to happen in that one cell, that one of the 4 billion blood capillary cells in the brain or the spinal cord. I often talk about the brain, but it's the brain or the spinal cord. The problem is, this isn't enough. You don't get a cavernoma simply by having this double mutation, the double hit for the Cavernoma gene. So what has to happen else? And in some of the earlier talks I've given, or some of the talks I did last year, I've talked about a third hit. And indeed, only in the seminar we had a month ago, I was talking about the way in which the microbiome, your gut microbiology, linked into Cavernoma through a substance, a molecule called lipopolysaccharide, and we thought about that as the third hit. And it's quite interesting that the person who did the work on the microbiome, which last week, which was revolutionary when it came out 3 years ago, it changed people's diets, is the same Mark Kahn has been the leader for this research, very active research. I mean, it's a very good place where he was working. It was about 300 yards from where I first met Pat. So, what is the extra? And the extra, which is the subject of this talk, is a gene called PIK3CA, and PIK3CA is known as a cancer gene. And we'll come to that in a moment. But that is in the normal cell is in the off position, and it can be mutated, a somatic mutation again, to form the on form. And when that happens, then you have what we call the third hit, and you can develop a cavernoma. But all of these hits have to be in that same cell, that one of the 4 billion blood capillary cells, in order for a cavernoma to form. And you can see that although you might be having hits all over your body at a moderately high frequency, the probability that you even have 2 hits is quite remote, and that you have 3 hits is even more remote. So it's a fairly rare event, but of course it does happen, as we know well, everybody in the audience. So that is known as a gain of function process, because the gene is off, as it were. It doesn't do its activation in the normal cell, but it is, when it's hit, it then gains its function and its ability to have all the effects we're going to talk about. So just a little bit about this gene, PIK3CA. It's a gene that's very commonly mutated in cancers, breast cancer, prostate cancer, other cancers. It's not the only way in which you get cancers in those cells, but it's a common one. As we said, it's a gain of function, somatic mutation. It turns on the gene, and the activation of that gene sends it into overdrive, and it stimulates cell proliferation and growth. And a little bit about how it does that in a moment. So in the research that we've just discussed here, the research that's gone in behind this, I'm making trivia of, in a sense. It's amazingly well-developed research from a group of people. You saw the number of people in the audience with a whole range of disciplines. And the one just to mention here, people donate their cavernoma when they're resected by neurosurgery to research labs, as they've done here. And those cavernoma, which then, when they're formed, have many, many thousands of cells in. But it's possible to identify or to take a single cell from that, something which is only about the size of a red blood corpuscle in rough round figures, and you can identify the genetics of that single capillary cell. And it was found, but so what was done was the researchers took these individual cells, and the way they collected those is the subject of another talk, really, and they then looked at the gene mutations in each of those cells, and did that for many cells. The Cavernoma has many cells, and you could identify those that came from the Cavernoma and ask which of those cells had which type of gene. And many of those cells, not all of them, but many of those cells, the majority of them, had two loss of function Cavernoma gene mutations, allowing the Cavernoma to form, and a single gain of function in the PIK3CA cancer gene. And the conclusion was that that was a requirement, almost certainly, for the very fast growth of Cavernoma that you get when you get a bleed in our common language terms, which gives rise to hemorrhagic stroke, to epilepsy or seizures, and to the whole range of symptoms that occur bigger or more when a Cavernoma bleeds. This is the link that has been searched for, that is the initiator, if you like, for a bleed. So, gain of function from the cancer gene, and we have to have the Cavernoma genes with their loss of function as well. Those three together, Cavernoma. I'm not seeming to get a Cavernoma for some reason. The third hit, Cavernoma. Well done. So I lose the pen sometimes for reasons that I'm not quite clear about. So that is the basic way that it works. So this is actually the penultimate slide, and this is taken very heavily from the article Angioma Alliance that came out this morning, that I saw it anyway. And so this gene, when it's activated, causes misregulation. You no longer regulate the molecules involved in cell metabolism, that's the general activity of the cell, and the cell's growth. And that happens via a molecule, an intermediate molecule, if you like, that's given the name MTOR. And the point about MTOR is that when it's there, it's overactive, and it's overactive in rapidly growing Cavernoma. And MTOR itself links into a sequence of reactions inside the blood capillary cells that we're not going to be concerned about tonight. And that is what is responsible for the rapid growth in the Cavernoma. It's also responsible for the rapid growth of shows that the growth of the individual Cavernoma is cancer-like, in the sense that this rapid growth also happens in these cancers that have these links. But the important aspect of this is that the response in Cavernoma, in the growth, has got a cancer-like ability of it, but the cells don't break away, they don't metastasize, and they don't spread in the way that malignant cancers do. This has got nothing to do with finding this out in this paper. We know full well that Cavernoma are not cancerous in the way that that is. And forgive me if I've already said this, my mind goes blank sometimes. Had this gene been discovered in Cavernoma to begin with, the one that is the one that triggers on the right-hand side of those diagrams, we would have probably called it a Cavernoma gene or something else. But the fact it was found in cancer cells before it was found in Cavernoma made it seem like a cancer cell. So just a sort of a quick word about cancers, for example. And one of the causes of breast cancer is that people have one of two what are called BRCA genes, which are sort of broadly equivalent in the development of breast cancer to the tight junction genes in the development of Cavernoma. In Cavernoma, you have tight junction genes and you have the PIK3CA becoming active. The result is a Cavernoma, and it's not cancerous. But when you have breast cancer formed from the BRCA genes that you might have, then the combination of whatever the BRCA genes do together with this is responsible for the other types of activity that occur in cancer. So it sounds very scary, and people, I'm sure, are very scared when they saw this saying a cancer-like gene responsible for the rapid growth of Cavernoma. But you shouldn't be worried from that. It is not a cancerous form. It is a way We don't want it to happen, it's absolutely true, but it's not going to be it's not going to become metastasize and create malignant cancer. And the best news is that there's a drug, rapamycin, it's got another name, and there are other similar drugs which target this molecule MTOR and block its activity. And that is used successfully with other vascular malformations. And experiments were done by the people who did this research, looking at the growth in mice, and rapamycin inhibits the Cavernoma growth in mice. And although that doesn't mean it necessarily happens in humans, it is very frequently the case. Mice are a very good what are called model for Cavernoma, because mice which develop Cavernoma, you find most of the time, whatever you find in mice is then found in humans when it's tested properly in humans. So the findings suggest a rather good thing. That is that rapamycin might well be a good drug for the treatment of this fast-growing Cavernoma, the ones that are going to cause all the problem and that are most commonly known to cause hemorrhagic stroke, seizures, and the variety of other conditions, the other symptoms that people with Cavernoma develop. So forget about the issues of cancer. Remember that we've got a new drug. It will have to be tested by randomized controlled trials, and I'm sure that will happen pretty soon. But of course, that's made easier because the drug is licensed in the States, I don't know about in Britain, for use in humans. And that is what happens in phase 1 of the randomized controlled trials. You've come across the 3 phases of the development of vaccines, of course, with coronavirus, and the phase 1 is not needed because we already know that rapamycin is a safe drug in humans. The question is whether it will then work to inhibit bleeds in Cavernoma. And that's the exciting thing that has come out of this paper. Now, let me say, this is the first of what undoubtedly will be a vast number of papers. I had a talk prepared for this evening that talked about the growth of Cavernoma, which I haven't given. And I haven't given it because it's been, in a sense, superseded. Well, it hasn't been superseded. The results of it are still accurate, and I may well talk about it. It's a very exciting development for how Cavernoma grow. But one of the unknowns in that talk was what happened to make these cells proliferate in a single Cavernoma. And now we know the answer. Whether it's the total answer, whether it's accurate or not, will still come. But this is the start of a new era. And that is the end of the talk.